Thank you, Ian. So my thesis is indeed on the stability of kelp and canopy-forming algae. It will consist of 2 chapters. In chapter 1, I'll discuss a case study I performed in Chile. And in chapter 2, I'll be talking about a review on the stability framework and a review on research. So let's dive right in. What are canopy-forming algae and kelp? Well, they generally belong to the order of the Laminariales or Fucales. They form vast kelp forests, which are defined as seascapes dominated by habitat-forming seaweeds. They can be found on all rocky shores of temperate and Arctic regions, and they dominate about 25% of our coastlines. They have a heteromorphic life cycle, which means it consists of 2 parts. The first one is the sporophyte, or the adult plant, in which they will release spores from its zooids on the blades. And these spores will then turn into the haploid stage of the gametophyte, with a male and a female gametophyte. Once fertilized, it will grow back into a sporophyte. They're very important for biodiversity. This can be in a direct way, as epiphytes live on the blades and stipes, but also rich communities of invertebrates live in the holdfasts. And also indirect, they enhance biodiversity, as the canopy acts as a refuge or a shelter for many fish, which also attracts predators. And due to physical engineering, they slow down the water, and in that way, and food retainment. For humans, they're also very important as they provide ecosystem services. These can be regulating, such as carbon sequestration into the deep sea or coastal protection. Provisioning, as they act as breeding grounds for many economically important species, or they're actually taken as food directly or used as fertilizer. And even alginates, a chemical, is taken from these seaweeds to use in various industries such as cosmetics. They also have a cultural importance, as they can be used in leisure time or attract tourism. Sadly, the kelp forests worldwide are threatened, and they have a global decline of 2%. This is due to a variety of drivers, which can work antagonistically or synergistically. A main driver is climate change, with an increase in sea surface temperature due to heat waves or the El Niño Southern Oscillation and others. Also, regime shifts occur. This can be also due to a variety of drivers, but generally leads to an increase in herbivory that can lead to barrens, or a transversion of the community in which canopy-forming algae are replaced by turf algae, which do not provide these ecosystems and do not have a high biodiversity. Also, anthropogenic effects can cause degradation of communities, such as harvesting of kelps. So then on to stability. Well, what is stability? It describes how an ecological entity evolves through time and space. This entity can be an individual, a community, a population, etc. Stability is multidimensional, and I use the stability framework for this thesis with a few of its aspects. So one of the important aspects is resistance, which is the ability to remain unchanged, or in other words, how well does the ecological entity cope with a disturbance. Recovery, which is the ability to return to a reference state after disturbance. So how well does it recover after being disturbed? And the last one, resilience, is the speed of the system to return to the reference state after disturbance, or how fast does it recover? And help understand the stability of the ecological entity, or just the ecological community. So in chapter 1, it's a case study on post-harvest recovery absence of Lessonia trabeculata. The central coast of Chile is dominated by the genus Lessonia. In Chile, they harvest about 10% of the world's alginic acids. And it used to be a mortality-based fishery in which beach cast kelps are taken. But at the end of the last century, it turned into an extractive fishery in which kelps are taken directly from the seabed. This can cause shifts in communities, and add onto that the threats I mentioned earlier. It can cause population degradation or even local extinction. So the study area was in Quintay, in the central region of Chile, where I used these 4 patches. It's a part of a harvesting experiment that was performed in 2016 on the kelp Lessonia trabeculata. This is a pristine kelp, or the dots, and this is a harvested patch, or the stars. And sadly, in 2020, we still see no recovery. So the research aim of this study was to provide preliminary results on the cause of recovery absence of Lessonia trabeculata over there. I did ask some questions, and these revolve around 4 important parameters of a kelp forest. the fecundity, whether that is impaired or not, the growth of the sporophytes, whether that's impaired or not, the sporophyte recruitment, whether that's impaired or not, and whether there's an increase in herbivory that can cause stabilization of the created barrens. So over to the results of fecundity. I used 2 assays. In the macroscopic assay, I went to look at the sori density. So I used IMBRSea, this is the sori. So I used IMBRSea to calculate the whole area of the leaf and then to see how much of that is taken up by the sorus. But the ANOVA showed no significant differences between the harvested and the non-harvested patches. In a microscopic assay, I just did a visual check if spores and gametophytes would be present. And as can be seen in the image here, the black arrow points to a spore and the red arrow points to gametophytes. We can see that they are present, and as such we can say that fecundity is probably not impaired. If you go down to growth, due to the pandemic, I could only do a proxy of growth, in which in situ we measured the holdfast diameter, the sporophyte height, the amount of stipes and the length of blades of sporophyte, but only the Welsh ANOVA on height showed a significant difference between patch 1 and harvested patch 1, and harvested patch 2. For further notes in this presentation, orange will always stand for harvested patches and green will always stand for non-harvested patches. I also tried to perform a new kind of experiment, at least for Lessonia trabeculata, in which rings that occur seasonally, like in dendrochronology, are counted. Sadly, the cross-section of Lessonia does not have this clear pattern as seen in Laminaria hyperborea, a kelp from Norway, and as such, this is not really useful. But overall, we can say that growth does not seem to be impaired. If you then go on to recruitment, we see that there is no recruit in the non-harvested patches. This can be due to the shading effect, in which the canopy covers or takes up all the sunlight and leaves not enough light for recruits to grow. But also very few recruits were found in the harvested patches. The recruits that were found were all clustered together in very small groups, which seemed to be some kind of refuge or a shelter, while the rest of the seabed of the harvested patches remained barren. This leads us to say that recruitment might be impaired. If you then look at herbivory, we counted the gastropod tegula and the sea urchins as a proxy for herbivory, and we found that twice as much urchins were found in the harvested areas and 10 times as much tegula. So some more detail of the urchins. The harvested patch 1 shows a significant difference from both non-harvested patches for the sea urchins, and the zero-inflated Poisson regression for tegula show that both harvested patches are significantly different from non-harvested patches. As such, we can say that there's a strong increase in invertebrate herbivory in the harvested patches. And then we will conclude this case study. So growth and fecundity did not seem to be impaired. Recruitment did, as there was no recruit in pristine sites and very few in the harvested patches. And there was a strong increase in invertebrate herbivory, which can stabilize the created barrens in the harvesting experiment, inhibit further recruitment, and cause the absence of recovery. Of course, some further research needs to be done on these data, and as such, I would collect more data. For example, on herbivore thresholds, at what point is there enough herbivores to cause an inhibition of recruitment? Also gather environmental parameters and long-term data sets to see whether this is, or whether the created barrens persist through time. Also check whether it's a local or a wider spread phenomenon. I would also perform a cultivation experiment in which gametophytes are cultured in the lab and see if they grow into adult and functioning sporophytes. And in situ, I would perform an herbivore exclusion experiment by placing cages in which herbivores cannot go into, and then see if recruitment occurs in the patches. On to chapter 2 then, the stability framework for canopy-forming algae and the review on research. During the case study, I found that there is inconsistent terminology use in publications. This can lead to confusion for readers or scientists. For example, sometimes when people talk about restoration, which is a man-help process, they actually refer to recovery, which is a natural process of recovery. There's also a lot of different definitions of kelp, including different taxa or species. Also, kelp forests and kelp beds are a discussed topic on what is what. But for this review, we'll be looking at stability. And I found that sometimes scientists refer to stability, while they actually only investigate resistance, or they refer to resilience, while they're actually looking at resistance and vice versa, etc. Therefore, to have a correct and consistent terminology, I propose to use a stability framework, which is with its earlier set aspects. Sorry, that was difficult. This can help understanding ecology, but also it's important to have correct terminology for good management policy. So some research aims and questions. I try to bring the stability framework to the field of ecology and at the same time provide an overview of the drivers in stability research on kelp. I did it also by some questions. Where is the research located? What taxonomic groups are investigated? And then 3 other questions regarding the stability, the drivers and their impact, and the study setup. But due to limited time in the presentation, I will not go into detail of those. So some general information. I used 146 publications that span about 4 decades. They're chosen based on their abstracts and keywords, and they all talk about disturbances and their influence on the stability of kelp and canopy-forming algae. And as you can see, there's an exponential increase in this type of literature. So where's the research located? Most research was done in the temperate Northern Atlantic, Northern Pacific, and Australasia. This is because these are regions with many universities and good funding opportunities. The reason that the Central Indo-Pacific is very low or last is that a lot of publications are made in the local language and therefore not used in this review. In this map, every publication that was used in the review is appointed by a dot, and it shows us some gaps worldwide where little research is performed, such as in South America, Africa, Siberia and Alaska, and also closer to the Arctic. This can be to a variety of reasons. One is limited funding, the other is the remoteness of these locations, and also the limited time with suitable weather for research. If you then look at the taxonomic groups, the 2 most investigated orders are the Laminareales and Fucales. This is also mainly due to 2 reasons. They have very widespread species and are very common in the world, and also they have many economically important species and are just better studied. So some conclusions for the review then. There's inconsistent terminology used regarding stability in publications, which can cause confusion. The stability framework, though, with its consistent terminology, can provide clarity. With the aspects of stability, can link research globally. They can help understand ecology and connect this to management and policy. The review also shows some understudied areas and genera, some more details regarding the research not included here. And as such, in total, the stability framework can improve research, policymaking, and conservation of kelp forests. So that leaves nothing else than to thank you all for listening, but also to thank my promoter and supervisor, Alejandro, for offering me the opportunity, to the Subelab team and all the ASIM staff for their help during my thesis, and of course my family and my friends for their support in pursuing my dreams.